What is this? Why, it's a contraption for Agarose gel electrophoresis. So, what the hell? Let's talk about Agarose gel. Agarose gel electrophoresis is a technique in which we can separate DNA fragments by size. It's really helpful for if you've done things like a PCR reaction you want to see works, if you want to see if you do like restriction enzyme digest, so you're cutting pieces of DNA and then you want to see the sizes of those pieces. So there's a lot of different uses of agarose gel electrophoresis, which we normally just like say like agarose, like I need to run an agarose, like or an agarose gel. So basically the basic idea, and then we'll get into the details more, is that the DNA has a negatively charged backbone. So you can use that to send it traveling through a gel towards a positively charged end. So this is electrophoresis. So using electricity to get the DNA to move. So the DNA is going to want to move because of that charge, but the gel mesh is going to make the DNA get all tangled up as it tries to travel through. And this means that when we turn off the electricity, so you stop having that force propelling the DNA through the gel, then and you look to see where the fragments are, the bigger pieces won't have traveled as far because they're going to get tangled up more. It's kind of like, I like to think of dropping a jump rope through a sea of basketball hoops. So the smaller fragments are going to be lower down, the bigger fragments bigger up, and farther up. And you can run like a size ladder of known DNA pieces of known size to get an idea of the size. So if you've been following along the past few days, this might remind you of PolyAcrylamide gel electrophoresis. So we looked at SDS-PAGE the other day, which is a similar technique in which we could separate proteins by size. There's also a technique called urea-PAGE where we use a gel to separate urea-coated RNA by size. So I'll show you in a minute a couple of the differences. So the main difference is that I'm showing here is what the gel is actually made of. So with agarose gel, it's made up of agarose, which is a sugar. And with PAGE, it's made up of these cross-linked polyacrylamide chains. So the cross-link means that these acrylamide chains are covalently bound. So they're really like tightly attached to one another. And so when you get it really hot or anything, it's not going to melt. You can also form a finer mesh than with agarose. But the really cool thing about agarose is it's kind of like yarn that you're knitting. The yarn like self-knits itself, but you're not actually making permanent covalent bonds. And so with agarose, you're not actually like cross-linking anything. You are there's no polymerization steps. It's just like a gelification. So what we do is we actually like make a solution of agarose and we heat it up in the microwave in a little flask to get it to dissolve, and then we could pour this gel and then it'll harden. It's really cool. And so basically it looks kind of like this. And so a key difference other than the type of thing that they're separating is that with an agarose gel electrophoresis, you have this horizontal slab. It's hard to see in this picture, but it's probably like half an inch thick maybe. And with an SDS-PAGE gel, you have a vertical format and you have this really thin gel that's actually, it's held in place by like glass plates or plastic plates. And so you're actually running it vertically. And so with either way, you're going from a negative charge end to a positive charged end. But with the SDS-PAGE, you do it vertically. And in this case, the gel is being held up between the plates. With the Agarose gel, you don't need plates, so you can just like lie it down horizontally onto this tray. And then, so for the visualization, so with the nucleic acids, for like the Agarose gel, you normally do a fluorescent stain, as I'll show you. Whereas with proteins, I'm showing you this was a Comassi stain, so a visual colorimetric or like color readout. But the basic idea is the same, that you have these things traveling through the gel and they're going to get slowed down more the bigger they are. But a key thing about the difference between SDS-PAGE and nucleic acids, other than what we've talked about, is that proteins are made up of amino acids, and there's 20 different common ones, and some of them are charged and some of them are not charged. And so your proteins are going to have various charges. Whereas with nucleic acids, so for DNA and RNA, you have a negative charge built into this backbone, and all of the so there's 4 letters and they all have the same negative charge. They're unique parts, so the nucleobases, those don't have any charge. So it's the backbone that has this charge, and all of nucleic acids have the same length to charge ratio. This is not the case with proteins, where you have various charges. And so for proteins, you actually have to give them the negative charge, which you can do with the detergent SDS. And the detergent also helps keep it unfolded and soluble. So like I said, you have this gel that's made up of this Agarose, and I just thought this was really cool when I learned how it actually works. So I have a post with lots more about that if you want to learn more about the Agarose. But yeah, it's really cool. So we don't need the SDS in our sample buffer, but we still need a couple other things. So by sample buffer, I just mean like the thing that we mix our DNA sample with before we stick it in the well and then turn on the electricity. A key thing is glycerol. So it just makes it heavy. So it doesn't just float out of the well. Even still, you want to make sure that you're like pipetting slowly. So you don't get like bubbles that just like pop out when you're trying to pipette it. So slow and steady wins the race. So the gels have these wells that you stick your sample into, and then you can run through. Cool thing about Agarose gels too is that you can kind of like reuse them. So if something has run through it already, you can still run something through it. And you can use like partial gels. So you can actually, like, if you only use half of a gel, you can cut it off and then someone else can use it. So in our lab, we normally like keep gels going. Like there's just like a common gel out there, and like if partial gel or whatever, and you just make sure no one else is planning to use it right then, and then you could use it, and they last for a pretty long time as long as you have buffer over them. Buffer is just like in this case, buffer is just like a pH stabilized salt water, so often TAE. And another thing that's in this in here is a tracking dye. So when the gel is running, you're going to see just this tracking dye. So we use this like Easy Vision, whatever. And so it has these tracking dyes so that you can see the progress of the run while it's going. This does not show you where the DNA is. It only shows you like an idea of where the dye front is. So, and then you can compare that to like what size it would correspond to in general. And also that's going to depend on the mesh size. So if you add more agarose, you get a tighter mesh. Less, you get a looser mesh. So a lower percentage of agarose is better for bigger things. A higher percentage is better for smaller things. So normally we do about a 1% gel for when we're analyzing PCR products for like a few thousand kilobases. So yeah, so the ladder here, you can see this is in like kilobase pairs. So on the right, you can see, sorry, I can't figure out how to make it so that you can actually see my pointer. But so on the right, you see like the visible dyes, and that's what you're gonna see when the gel is running. On the left, you have the, what happens when you have a fluorescent stain. So in this sample buffer that we use, it has a built-in fluorescent stain. But you can also fluorescently stain afterwards by soaking the gel in a fluorescent stain, or you can make your gel with the fluorescent stain. So what do I mean by fluorescent stain? So fluorescence is basically where you shine a light of one wavelength at something and it shines back at a different wavelength. A couple common nucleic acid stains we use are ethidium bromide and DAPI. So DAPI, they just bind DNA in different ways. So ethidium bromide's gotten a really bad rap over the years, but because what it does is it actually like intercalates in between stacked bases of DNA, so it like wedges in between them. And this can cause problems if this was like in your body, because it can cause the like DNA copying machinery and stuff to make mistakes. But it turns out that ethidium bromide is really not, you'd have to have like basically drink a ton of it in order to have any negative effect, and so-called like safer stains aren't really much safer usually. So yeah, just follow good lab technique when using it. Wear gloves, wash your hands afterwards, the usual stuff, no matter what stain you use. Yeah, so something to keep an eye out for as well is that, so with protein, so with SDS-PAGE, one of the things that SDS does, in addition to add the negative charge, is it unfolds the proteins so that they stay linear and you separate them based on their length instead of like their shapes, which would get in the way of them moving through the gel. So with DNA, usually, so if you have like linear DNA, it's not an issue. But if you have like a plasmid, so a circular piece of DNA, it's going to travel weirdly. So basically, if you have like, if the plasmid is super coiled, so this is kind of like if you have a phone cord, like one of those curly phone cords that is all coiled up, we call it supercoiling. And basically that makes it like a really compact little ball that can just like whiz through the gel. So that's going to travel faster. If you have a plasmid that's nicked, so it's not super coiled, but it has like one strand. So basically when it's super coiled, it like, cause it has all this tension. So if you have like a nicked plasmid where the tension is relieved, it's going to travel slower than the linearized version, because it's kind of just like this weird sluggish like pancake thing. Not pancake, but it's kind of like an awkwardly shaped and hard to get through the gel, whereas the linearized can kind of like move snake-like through the gel. And yeah, so this is just showing you like what you would see in the case of nicked, linearized, or supercoiled DNA. And yeah, so that's basically all I wanted to say about Agarose gel electrophoresis, and there are more pictures and text and stuff on my blog. So good luck. Oh, and make sure that you run to red. You always want to make sure that you're putting the positive end of the gel where you're loading the wells, or sorry, when you're, no, yeah, where you, at the bottom of the wells, and then the negative end where you're loading so that the DNA gets repelled from the start to the end. And since it already has that negative charge, you're good to go without SDS. But you need that buffer to hold it down and give it color. So happy experimenting.